Hello students, I am Fazle Amin and today we will continue the last and the final lecture about the extraction methods, and these extraction method includes maceration, percolation, large scale extraction. So today I'll discuss one by one in detail the maceration process, the percolation, the large scale extraction. Objectives: the main objectives is to learn about the different methods of extraction. These includes the maceration, percolation, and large-scale extraction, its purpose and process. After this lecture, I'm sure the student will be able, all you will be able to know about the basic concept, purpose, and the various steps involved in each method of extraction. There are various methods of extraction in pharmaceutical field, which include maceration, percolation, infusion, decoction, digestion, and the large-scale extraction, which is also called extraction battery, which is a continuous extraction process. So today I'll explain one by one in detail. So the first one is maceration process or maceration method. The term maceration comes from the Latin word miserere, which means to soak. So this is the simplest method of extraction. The process. So number one, in this method, a definite quantity of properly comminuted, crushed, or moderately coarse powder of the crude drug of plant is placed or soaked in a wide container containing menstruum and close the vessel tightly. Then the vessel is agitated repeatedly for over the period of usually ranging from the 2 to 14 days at a temperature at about 15 to 20 degrees centigrade, or until the crude cellular structure is softened by absorbing the menstruum. And lastly, the soluble constituents are dissolved and extracted out with or without force, and finally filtered and use it. The purpose of maceration. The purpose of maceration process. The maceration process is used for those drugs containing little or no cellular material such as benzoin, or we can say that the maceration process is used for those drugs which are quickly or almost dissolved completely in menstruum. Uh, so so we can say the main purpose of this method is to just soak the crude drug in a menstruum, and the extractant is ready to use. So it is the simplest method amongst all the extraction process or extraction methods. The second method is percolation, and percolation is the combination of two Latin word per, meaning through, and colere means to strain, which is described generally as a process in which the comminuted drug is placed in organized form for extraction of soluble constituents by slow passage of menstruum through a column of narrow cone-shaped container which is open at the both end, called the percolator. The percolation method is mostly used for the preparation of tincture and fluid extracts. Percolation on a small scale generally involves the use of glass percolators of various shape for extraction of small amount, usually up to 1000 grams of crude drug. And the percolator used for the large scale generally made of stainless steel or glass lined large metal vessel available in various sizes, even 6 to 8 feet in diameter and 12 to 18 feet high. So this is a diagram of the percolator. So in this slide, I'll give a detail of the processing step or the procedure. The first and the most important one is the preparation of the dry crude drug for the percolation, that is powdering and the moistening. Number two, the packaging the percolator, which means that the crude drug is packed in organized form inside the percolator. Number three, the period of maceration, which means that the time of soaking in a menstruum. Number four, percolation and collection of percolate, which means that after soaking, additional menstruum is added and the flow is adjusted by collecting the percolate from the bottom of the percolate. And lastly, the adjustment of concentrate of the percolate, which means that the required concentrate is achieved by equilibrium or constant flow, that is, addition of the menstruum and the collection of the percolate or extractant. This, this is a procedure steps. At least six steps are required in percolation process. In this process, the crude drug is packed in a percolator in a well-disciplined manner and moistened with the appropriate amount of the menstruum. The top of percolator is closed and allowed to stand for approximately 4 hours. An additional menstruum is added to form a shallow layer above the mass, over the crude drug. Number three, the mixture is allowed to macerate in a closed percolator for 24 hours. The outlet of the percolator at the bottom is opened and the extractant is allowed to drip slowly. Number four, as it drips slowly, the additional menstruum is added as required, and the menstruum is added until the last trace of active constituent is isolated. Number five, the mark is then expressed or squeezed to drain all the extractant, and lastly, the extractant is clarified by filtration or by standing, followed by sedimentation or other using equipment like centrifuge machine containing filters. The purpose of the percolation. So the main purpose of percolation method is to extract the maximum required active constituents. And secondly, we can also treat the crude drug with different selected solvent, that is, aqueous soluble and fat soluble constituents of the crude drug can be treated with different menstruum using different condition and methods. But remember that only one menstruum should be used at a time. The large scale extraction, when the large quantity of crude drug need to process or the efficiency of the extraction is improved. So in both cases, we prefer large scale extraction. In large scale extraction, you usually refer or call extraction battery or the counter current extraction, as we discussed in a previous lecture. The process, as discussed in the previous slides, a huge vessel or tanks are used when a large scale extraction is required. So stainless steel or glass lined large metallic vessels are mostly used, having size 6 to 8 feet in diameter and 12 to 18 feet in height or even bigger than that. In this technique, the vessels are arranged in such a manner that the menstruum is added to the container at any time and can be taken from any vessel at any time. So in this method, the extraction process is improved by arranging the vessel. The solvent is continuously circulating through the crude drug repeatedly and continuously. In this multiple stage extraction, the menstruum is distributed through spray nozzles over the surface of the crude drug from the top of first vessel by powerful pumps, and when the sprayed menstruum reach to the bottom of the vessel, so automated pumps pump from the bottom of the vessel to the inlet of the second vessel and so on. And in this way, repeat the cycle by collecting the extractant from the last vessel to the first vessel and circulating continuously until the required concentrate is achieved. So here we can see the stainless steel vessel, and here at the bottom of each vessel is a powerful pump which transfer and spread the menstruum on the top. Here you can see on the top of the next vessel and so on. The purpose and scope of the large scale extraction, when the large quantity of crude drug need to process or the efficiency of the extraction is improved. So in both cases, we prefer a large scale extraction. It is also important when the conventional extraction method, that is in the normal percolation or maceration, when the percolate is not of the maximum concentrate, so large scale continuous extraction devices like extraction battery are you get the maximum concentration in the short time of period. And the aim and the purpose of large scale extraction is not only to minimize or to control the wastage of menstruum, but also control the hazardous substances, and it is also an environment friendly. These books will help you in reading the extraction process. The lecture is taken from these three books. This is all for today. Thank you very much.